Imagine trying to understand a movie by watching a single frame every 5 minutes. You would catch a glimpse of important moments, but the context of the story would be lost entirely. Today, this is the prevailing way we study molecular biology. We only get snapshots, fragments of information, using conventional methods like proteomics or genomics that shows only the outcome, but not the process. We're left guessing, trying to reverse engineer the full picture of how life works, but most of the details are missing. To solve this problem, the field of molecular recording arose, a subject that is little known by the general public and the scientific community alike, as their survey suggests. Molecular recording systems can turn cells into their own chronologies by recording events occurring inside them, such as the presence of certain ligands, nucleotide sequences, or even proteins. However, most recording systems are developed on DNA, which makes accessing and reading recorded information time-consuming and requires cell lysis. Moreover, due to the nuclear localization of DNA, it is difficult to rapidly record events in the cytoplasm, but through the cell membrane. Think of it like how computers store data on hard drives for long periods of time. Accessing this information can be slow. To address the challenges of DNA-based recording, we drew inspiration from the way computers handle data that needs to be accessed quickly. Looking at how RAM, random access memory, allows computers to rapidly store and retrieve data, we realized that RNA, as the cell's messenger, could play a similar role in molecular recording. Therefore, we would like to present to you our iGEM 2024 project called ProgRAM, Programmable RNA Access Memory, a new generation of molecular recorders. Here's how we did it.
Our molecular recorder has two main functions, writing and reading. First, we needed a robust and reliable system to write on RNA. To achieve this, we turned into Repair, an established RNA editor for programmable A-to-I replacement. Specifically, we employ a dead Cas15b enzyme from the Prevotella species coupled with an RNA deaminase domain ADAR. Upon sensing a cellular event, a gRNA is expressed, which guides the dCas15b ADAR complex to its target. When on site, ADAR catalyzes the deamination of a single adenosine to inosine, which is recognized as guanosine during translation. This process enables RNA to serve as a dynamic recording tape, where each modification represents a logged event. To validate the editing system, we developed a nanoluciferase restoration assay by introducing a point mutation which created a preliminary stop codon in the luciferase gene. We were able to measure luminescence only if the repair system successfully edited the introduced adenosine, restoring luciferase activity. We could confirm the system's reliability, and our assay optimization showed that the 5-fold molar excess of the editing complex gave the best results, guiding future experiments. Having successfully tested our editor's efficiency, we started designing the heart of our system, the recording tape itself. ProgRAM enables precise molecular recording for a logic embedded within its tape design. By engineering overlapping gRNA binding regions, we ensure that each deamination is performed sequentially, allowing accurate event tracking. For fast and accessible readout, the overlapping target regions were equipped with successive start codons in different open reading frames. As a result of a deamination, a start codon is inactivated and a +1 frameshift occurs, allowing the expression of a reporter in one of 3 frames, depending on the recorded state. We carefully designed the deamination targets and surrounding nucleotides to ensure both high editing fidelity and efficient translation initiation. Using a deterministic in silico approach, we generated and selected 5 tapes ensuring structurally stable gRNAs with high on-target binding affinity and minimal local or global off-target potential. To analyze the efficiency of our tape switching from an unrecorded state denoted as state 0 to a recorded state 1, we designed another N-loop-based assay. For this, we cloned our tape with an N-loop gene, so that the correct reading frame encoding luciferase is translated only when the first start codon of the tape is deaminated, representing the recording of a single event. Synthesized tapes with pre-deaminated start codons were used as controls. Our experimental results showed that the recording process is most efficient in tapes 3 and 5. Having confirmed single-state switching, how can we now implement an in vivo readout system that tracks the recording of multiple events?
For that purpose, we incorporated a 3-color fluorescent protein readout system into ProgRAM. This traffic light system allows for real-time readout of multiple states while keeping the cells intact. Each state recorded on our tape is visualized by expressing one of three fluorescent proteins, MRFP670 M-scarlet3, or MTEC-BFP2, each corresponding to a different reading frame. These XFPs were selected for their non-overlapping emission and excitation spectra, as well as maturation time and fluorescence intensity. A significant challenge in translating these fluorescent proteins from a single construct has been the optimization of codon usage while avoiding premature stop codons across all three reading frames. Although we explored commercially available solutions for this sequence design task, none could handle multi-frame codon optimization while excluding several custom sequence motifs. This led us to create our own sequence optimization tool using an evolutionary algorithm based on a multi-objective optimization approach. Our algorithm mimics natural evolution, starting from a population of 500 back-translated DNA sequences. Variations are introduced through mutation or mating, where parts of sequences are exchanged through crossover. We then applied selection pressure over 300 generations, using metrics such as the codon adaptation index, frequency of optimal triplets, and exclusion of stop codon, among others. When analyzed on a single-frame basis, the quality of the sequence generated by our algorithm rivals that of existing tools. With expert guidance, we fine-tuned our codon optimization algorithm, carefully selecting the best parameters and quality controls. Experimental validation demonstrated the effectiveness of our algorithm, as we were able to individually express functional fluorescent proteins from the generated sequences. In addition to optimizing the fluorescent protein sequences, we added different sequence elements to tune the performance of our tape. We included a total RNA expression control by co-expressing EUNAG fluorescent protein in all three frames. Within the coding sequence, all elements are separated with optimally arranged ribosome skipping sites to maintain seamless and efficient translation. Beyond the coding sequence, our human practices work led us to utilize UTRs to further optimize reporter expression. We selected a highly stabilizing 3' UTR and 5' UTR that enhances ribosome binding at the cap. Additionally, we generated a synthetic UTR using a generative neural network combined with a gradient descent algorithm to ensure high transgene expression. With all the parts in place, we set out to conduct the proof of concept validation of ProgRAM. To achieve this, we introduced the repair writer system, along with various tape recorders and guide RNAs into HAC293T mammalian cells. Starting 24 hours post-transfection, the cells were imaged every 12 hours using a high-content imaging system. When testing single-state switching in cells expressing only gRNA1, we observed fluorescence from both MRFP670 and M-scarlet3, while the MTEC-BFP2 signal was absent. However, by 72 hours post-transfection, fluorescence could be detected in the MTEC-BFP2 channel, suggesting slight ribosomal slippage or non-specific gRNA binding, leading to undesired expression over time. In samples where both gRNA1 and gRNA2 were provided, allowing for 2 consecutive state switches, a low MTEC-BFP2 signal was detected at 24 hours. By 72 hours post-transfection, the signal had increased, suggesting that the recording could successfully progress to the third reading frame, as expected. To build on our preliminary proof-of-concept data, we plan to expand testing to more tape designs and conditions, while also refining our image acquisition and evaluation protocols. We aim to present and discuss additional experimental results at the Grand Jamboree.
One of the key advantages of ProgRAM is its modularity, allowing core components and add-ons to be combined and customized. Some add-ons that we explored as a part of our human practices efforts include integrating the tape into a viral replicon for improved longevity and robustness, inducible Cas13 editor expression through chemical or optogenetic controls, optimizing editor degradation rates for enhanced temporal resolution, and incorporating aptamers or exosomal signals for specific localization. This makes ProgRAM highly adaptable for various future applications, providing multiple insights on the use of our system, allowing it to be seen as a prominent tool in areas like gene therapy monitoring and real-time tracking of sequential gene expression. More broadly, these efforts come from a deep reflection on our scientific practices through novel socio-technical integration research protocols, which we made available for the iGEM community to use. For us, this is the heart of iGEM, not thousands of isolated projects, but an evolving, collaborative initiative. However, accessing this database of knowledge becomes increasingly difficult. With over 4,470 wikis to read through, current tools offer only surface-level details, like the depth of the work done by previous iGEM teams. Here, we developed the iGEM Data Explorer, a custom AI-powered tool that offers an accessible, interactive way to explore the scientific content hidden in iGEM wikis. Currently, as part of our proof of concept, data from 2019 wikis has been collected, but the vision is to expand the tool into all-encompassing resource for every iGEM here. Whether you're curious about which projects use Cat13 or seek guidance on starting a project with cyanobacteria, the iGEM Data Explorer can help you find relevant scientific work done by previous iGEM teams. Acknowledging AI's evolving role, we also organized an interdisciplinary panel discussion on the future of AI in modern biology. Broader educational efforts included events like Girls' Day, iGEM School, Bioinformatics Day, Dome and LMU Open Days, for which we created 3D printable puzzles, the central dogma of molecular biology. Our project wouldn't have been possible without the support of our PI, guidance from our instructors, and valuable feedback from stakeholders. We also thank the institutions and companies for their financial support. Your help has laid the foundation for ProgRAM to illuminate the new details that shape the future of molecular recording.